Okay, so we will continue with photosynthesis today, and we will be talking about the light reaction. We will be talking about the light reaction of photosynthesis. Okay, now I have already mentioned this to you that the light reaction is the photochemical phase. It is the photochemical phase. What does it involve? It involves, number one, the light absorption. The second thing that it involves is water splitting. And the third thing that it involves is oxygen release. Now, very important in the light reaction phase is that it leads to the synthesis of two intermediates, which is ATP and NADPH. And this ATP and NADPH, these are going to be required in the dark phase. These are the raw materials for the dark phase. Okay, now of course several complexes are involved in this process. Now what I want to mention over here is that I have already said that this entire thing occurs in the chloroplast, and the chloroplast, it has certain pigments which are known as chlorophyll. Okay, now the pigments, they are organized, these pigments are organized into two discrete light harvesting complexes. There are two discrete light harvesting complexes which I'll call LHC. Now there are two distinct light harvesting complexes. One is known as photosystem 1. This is photosystem 1, and the other one is known as photosystem 2. Now they are being named in the order of their discovery. Photosystem 1 was being discovered early and photosystem 2 was being discovered late. Okay, now each of these light harvesting complex are made up of hundreds of pigment molecules which are bound to protein, and each of the photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system which is also known as the antenna. Okay, which is also known as the antenna. Now these pigments, they help to make the photosynthesis more efficient. See, the chlorophyll is going to absorb the light energy. It is directly involved in photosynthesis. However, the antenna pigments, they help to make photosynthesis more efficient by absorbing different wavelengths of light, because they have a fixed wavelength. So all of these antenna, they are going to clustered the different wavelengths of light, and they are going to sequester it to these photosystems, to these reaction center. A single chlorophyll A molecule forms the reaction center. There is a single chlorophyll A molecule which forms the reaction center. So you have a reaction center here, and this is the other reaction center that you have. The reaction center, now this reaction center, it is different in both photosystems. In photosystem 1, the reaction center chlorophyll A, over here the chlorophyll A that you have has an absorption peak at 700 nanometer. Okay, the photosystem 1, in photosystem 1, the reaction in photosystem 1, in the reaction center, the chlorophyll A has maximally, it absorbs at 700 nanometer, and that is why the photosystem 1 is also known as P700. Remember one thing, this is the maximum range of absorption. It absorbs maximally at 700 nanometer, and hence it is known as P700. While in photosystem 2, in the reaction center, the absorption, the chlorophyll A has an absorption has a maximum absorption at 680 nanometer, and hence it's known as P680. So I repeat, there are two light harvesting complexes, photosystem 1 and photosystem 2. In photosystem 1 there is a reaction center, in photosystem 2 also there is a reaction center. There are chlorophyll A molecules over here, and of course there are antenna pigments over here also. Chlorophyll A directly involved in photosynthesis, and the antenna pigments, they help to broaden the range of absorption of light by the chlorophyll. Over here, it absorbs maximally at 700 nanometer, hence it is known as P700. Over here, it absorbs maximally at 680 nanometer, and hence it is known as P680. Now, in my previous lectures, I've already mentioned that for ATP synthesis, there are basically two steps. Number one is electron transport chain, and the second is, of course, due to the electron transport chain, a proton gradient is created, and it's on the virtue of this proton gradient that ATP finally gets synthesized. So there is an electron transport chain, and then there is phosphorylation of ADP into ATP. Now, since this occurs in presence of light, we call this photophosphorylation. We call this photophosphorylation because this ADP to ATP, this occurs in presence of light. Now, as I have already mentioned, there are two photosystems that are involved over here. There are two light harvesting complexes over here. So you have photosystem 2 over here, you have photosystem 2 over here, and you have photosystem 1 over here. So this is photosystem 2 over here, and this is photosystem 1 over here. Okay, what happens is that both of these photosystems, now I have also mentioned that this photosystem 2 means P680 and photosystem 1 means P700. Photosystem 2 means P680, photosystem 1 means P700. Now both of them, they absorb photons. They absorb photons. So in presence of light, what happens is that their energy level increases. Their energy level increases. Okay, like this. So if I measure their energy levels, if this was their ground state, their energy level increases. Normally, what happens is that if a substance, its electrons get excited to the higher energy level, then what happens is that it returns back. Once it gets excited, and then it may lose the energy, and then it returns back. But over here, what happens is that you have an acceptor of these electrons over here. The energy gets excited, the electrons get excited from here, and then what you have over here is you have someone who can accept the electrons over here. In this case, you have coenzyme Q, who acts as the primary electron acceptor here for photosystem 2. And then you also have a primary electron acceptor of photosystem 1 who accepts the excited electron. So the excited electrons are being accepted by CoQ over here, and the excited electrons are accepted by by the primary electron acceptor 1 over here. Now when the electrons are jumping up in the higher energy level, and when someone else is accepting it, obviously an electron hole is going to be created over here. I repeat, obviously an electron hole is going to be created over here. So there is a hole of electrons, there is a lacuna of electrons over here. Now what happens is that the CoQ, it donates the electrons to plastoquinone. It donates the electron to plastoquinone, and then it donates the electron to cytochrome B6. It donates the electron to cytochrome B6, and this cytochrome B6, it donates the electron to cytochrome F. It donates the electron to cytochrome F, and the cytochrome finally donates the electron to plastocyanin. So in case of photosystem 2, what happens is that the electrons are given to coenzyme Q. From there, the electrons move to plastoquinone, from there to cytochrome B6, and from there to cytochrome F, and finally from cytochrome F to plastocyanin. Please note over here, what happens is that while the electrons are being, are moving from cytochrome B6 to cytochrome F, the protons are being pumped inside. The protons are being pumped inside. I hope you remember in the last lecture I had said that the protons are moving from the stroma to the thylakoid space over here. Okay, so the thylakoid space, it is rendered more acidic, and while they are trying to come back over here, hence ATP is being produced from ADP and PI. Okay, so it is at this step that the ATP gets produced. Over here the ATP gets produced. Okay, and the electrons are moving from photosystem 2 to coenzyme Q to plastoquinone to cytochrome B6 to cytochrome F and finally to plastocyanin. Similarly, the electrons which are excited over here, they are being rendered to ferredoxin. They are being rendered to ferredoxin, and over here you have another enzyme which is known as ferredoxine NADPH oxidoreductase. So you have this ferredoxin NADP oxidoreductase, and what it does is that it transfers the electron from ferredoxin to NADP, and NADP finally gets converted to NADPH. Fine, it gets converted into NADPH. So this is how in this entire procedure of phosphorylation, ATP is produced, that is the first byproduct, and you also have the NADPH production. Okay, now I have already mentioned that here you have a hole that is created in photosystem 2 and you also have a hole that is being created in photosystem 1. What is interesting is that you have water molecules. You have water molecules and there is a splitting of water molecules. There is a splitting of water molecules. If I consider 4 H2O, then there are 4 OH minus, from where you get 4 electrons, and these are being donated. These electrons are being donated to photosystem 2. These are being donated to photosystem 2. So in presence of since there is a snatching of electrons, see over here, since there is a hole that is being created, photosystem 2, it snatches the electron from water, and while it is snatching the electrons from water, the water gets split into OH and into H+. This H+ is basically required over here for the conversion of NADP into NADPH. And this is how the hole that was being created in photosystem 2 is being rendered fine. Okay, and now there is a hole in photosystem 1 too. How is this electron hole in photosystem 1 subsided? What happens is that the electrons which were being donated to plastocyanin, that gets donated over here. So apparently it seems that water, now let's have a look at the entire scheme. See, what happens is photosystem 2 gets excited, photosystem 1 gets excited. Each of these have their primary electron acceptors. From over here it's CoQ, over here it's primary electron acceptor 1. From CoQ it moves to plastoquinone, to cytochrome B6, to cytochrome F, to plastocyanin, and finally it appears as if from plastocyanin it moves to photosystem 1, while the electrons from photosystem 1, they move to ferredoxin, and then ultimately it is used for the reduction of NADP into NADPH, and the electron hole that is being created over here, that is being subsided by water molecules. So apparently it seems that the electrons from water are moving from photosystem 2 to photosystem 1 and ultimately to NADP. And in the procedure there is the production of ATP and then there is the production of NADPH. And since the scheme somehow looks like a Z, see, it looks like a Z, this is known as the Z scheme of photosynthesis. It is known as the Z scheme of photosynthesis, and this is also known as non-cyclic photophosphorylation. It is also known as non-cyclic photophosphorylation. I repeat, ultimately it seems that the electrons from water, they move to photosystem 2, it gets excited, there is an electron transport chain which leads to the production of ATP that moves to photosystem 1, and then it finally moves to NADPH. It seems like this, but actually it is not the case. Individually, photosystem 2 and photosystem 1, they get excited separately. It does not depend on each other. Okay, but yes, the whole fill up depends on each other. The hole that's being created in photosystem 1 definitely is being subsided by photosystem 2. The electrons of photosystem 2, and the hole that is being created over here is being rendered by the electrons from water molecule, and this is known as non-cyclic photophosphorylation.